Today, Andre and I will be doing a demonstration of the Red Cabbage Indicator lab. Some materials that we will need for this will be, of course, safety goggles, a red cabbage, a knife, test tubes and a test tube rack, a hot plate, a couple of 250 milliliter beakers, a funnel, 10 milliliters of bleach, detergent, vinegar, and Windex, one 10 milliliter graduated cylinder, distilled water, and a pipette. Here we have a couple of 250 milliliter beakers, and now we need to slice up the red cabbage. Yes, we need to get the red cabbage into pieces. Now we can take the slices of the red cabbage and we can put it into a beaker of boiling water. And we do this because we need to remove the pigment that is inside of the red cabbage. So we'll need to stir the red cabbage that's in the boiling water for about 10 minutes. So the reason we're doing this is to extract the water-soluble pigment that is found in the red cabbage, which is called anthocyanin. And this pigment can be used as a pH indicator. Anthocyanins are present in many plants, but anthocyanins are complex molecules which undergo structural changes due to changes in hydroxide and hydrogen ion concentrations. The structural changes in the molecules react differently with the photons and exhibit different colors. So here you can see the differences in the molecular shapes after they've been in acid and bases. And here are the different colors that a red cabbage changed to in different solutions. Here the red cabbage has been fully soaked in the boiling water and it's ready. So now we need to remove the red cabbage juice. We need to make sure that we don't get any of the actual red cabbage into our beaker. And about 250 milliliters will suffice for this experiment. The pH of the solution is it's about 7. So first we're going to test bleach, and we're going to dilute the bleach, 10 milliliters of the bleach in 10 milliliters of water. So here is that solution. And let's take the pH of this. The pH of this is around 9.7. So now we are going to add the red cabbage solution into the bleach solution, 10 milliliters of the red cabbage. So as we pour it in, it is turning to a yellow-orange color. We can see this a bit more clearly. Yes, a yellowish color, which is what it should be, as told by the table. Next, we're going to test the detergent. And the pH of the detergent is 9.3, about 9.3, 9.29. Now we are going to add the red cabbage indicator. Doesn't really matter how much we add. And you can see the solution turning pink, turning a purple color. Next we're going to test the ammonia, and we're going to dilute this as we did with the bleach. So it's 10 milliliters of the ammonia, 10 milliliters of water, and the pH is around 10.67, about 10.7. Now we are going to test this with the red cabbage indicator. And as we pour it in, we can see that the solution turns a blue-green color. You can see, and over on this table we can see that the blue-green color is exactly what a solution of around 10, a pH of 10 should be. Next we're going to test the vinegar. The pH of the vinegar is around 2.73. We're now going to put the red cabbage indicator into the solution, and it turns a bright pink color. You can see bright reddish pink. This is and as we can see over here, a pH around 2, that's what this should be. Thank you for watching this demonstration.